my name is Nur Fatiha binti Muhammad Nasir, and today I'm going to carry out an experiment about the effect of temperature on fish respiration. So the objective of this experiment is we want to identify the effect of water temperature on the respiration rate of a fish. Respiration is the way organisms exchange gases with the environment. Aerobic respiration occurs when oxygen is taken into the body and sent to all its cells. It is then used to break down food for energy. All organisms experience respiration differently. Lungs, gills, trachea, and integument are all different structures equipped for making respiration possible. Respiration in fish takes place with the help of gills. Gills are tissue made up of feathery structure called gill filament, providing a large surface area for exchange of gases. A large surface area is crucial for gas exchange in aquatic organisms as water contains very little amount of dissolved oxygen. There are two hypotheses in this experiment. The first is the fish breathing speed faster when it is in the normal temperature water. The second one is the fish breathing speed is lower when it is in the cool water. So this is the materials that we need in this experiment. The first is 200 ml of tap water. Second is 200 ml of cold water. And we need a fish. First, put the fish in the beaker that contains 200 ml of tap water. Next, we observe and record the number of breaths every minute by counting the number of times the fish opens and closes its mouth for 5 minutes. The next step is we put the fish in the beaker that contains 200 ml of cold water. Observe and record the number of breaths every minute by counting the number of times the fish opens and closes its mouth. So this is the result for the experiment that has been carried out. I found that my data supported my hypothesis. It is true when the fish are put in warmer water, their breathing speeds up, and when fish are put in cooler water, their breathing slows down. At normal temperature water, maximum respiratory rate of the fish was recorded for the average 46 times per minute. In 5 minutes, it was found that the numbers of the fish breath is 380 times. But when the fish was exposed to low temperature, which is in the cold water, its respiratory rate was found to be decreased from 76 to 51 per minute in average. And in 5 minutes, the number of the fish breath is 255 times. Hence, in the present study, the impact of different temperatures on the freshwater fish show increase in respiratory rate at normal temperature, while at cold temperature, it can be observed that there is a decrease in respiratory rate. Therefore, this indicates that with increase in temperature, the metabolic rate of fish was also increased, which raised the oxygen demand, but reversibly, it decreased oxygen solubility in water. Hence, rise in atmospheric temperature due to normal variation will directly influence the water temperature and ectothermic fishes. It also shows that temperature acts as growth rate modulator of the metabolic capacity of fish. Other factor that affect the rate of respiration in fish is the concentration of the oxygen. The shallow water fish need high level of dissolved oxygen in water to undergo respiration. Therefore, when the concentration of dissolved oxygen in water is low, the rate of respiration in fish will also decrease. This study indicates that the increase in temperature of water, the respiratory rate of fish was also increased. This shows stress on the metabolic process of freshwater fishes. Thus, there is need of maintaining the environment temperature for preservation of local freshwater fishes for a long time.